Awesome. Very excited to next present Joakim Clifford, who is a student at the Denmark Technical University with our longtime collaborator, Dr. Morten Nielsen, who will also be defending his thesis, I believe, very shortly. So he's very kind to join us today and present the B cell epitope prediction tools. So thank you so much, Joakim.
Thank you for that introduction. I'll just share my slides. Awesome. Um, yes, today I have the privilege to talk about B-cell epitope prediction tools on IEDB. And we'll start with this is an outline of the topics we'll be talking about. So we'll start with a recap of B-cell epitope biology, just to bring everyone up to speed of what we're trying to predict with these tools. And then we'll get into two types of prediction tools, sequence-based and structure-based. And then we'll have some examples, and then we'll have some up-and-coming tools and some conclusions. So, as you know, B cells are these immune cells that we have in our body. And on the surface of these cells, there is a protein called the B cell receptor, although it is more commonly known by its soluble form, which is the antibody. And the molecule that it interacts with, we call that the antigen, but more specifically, the interaction site of the antigen, we call that the epitope. And that is what we're trying to predict with these tools. Um, and one antigen can have multiple epitopes from different antibodies. And our gold standard for data for understanding these interactions are structures of antibody and antigen complexes, which we can determine using X-ray crystallography or more recently cryo-electron microscopy. So here we have one such example where we have hemagglutinin from influenza A virus, which is complex with an antibody. And you'll notice it's just the variable domains of the antibodies. Um, you typically don't have the entire antibody crystallize due to experimental constraints. But this gives us enough information to um, see the epitope, which we're trying to predict. And uh, just so you know, the amino acids on the antibody side, which interact with the epitope, we call that the paratope. So the epitopes that we're trying to predict, they come in two flavors. They're either discontinuous or linear. Linear means that if you look at the epitope in the amino acid sequence, they'll be next to each other. And discontinuous means that they're not necessarily next to each other in the primary sequence, but the fold of the protein brings it together to in a spatial proximity to form a B-cell epitope. And if you just do some statistics looking at all the cell structures we have, um, you can find that the vast majority of B-cell epitopes they are discontinuous. That concludes the recap. Hopefully, that will make the rest of the presentation make more sense. Um, so going into the tools, we'll start with the sequence-based tools, and these are typically used to predict linear epitopes. But before we get into the tools, I think it's important to talk about when you should use them, because the gold standard is, of course, actual experimental data. So the first thing you should do is check whether or not there is epitope data for your antigen of interest. And even if you can't find something for your antigen of interest, then maybe you could find data for an antigen that's very similar, and then those epitopes can be mapped to your antigen. Another use case is if your antigen is understudied, so you might only have one epitope discovered for your antigen, and then you want a more comprehensive overview of other possible um, binding sites for antibodies. And that's where these prediction tools also can be helpful. So, um, Yeah, so for predicting these epitopes, we actually do have some idea of the physical chemical properties of what epitopes are, which residues are epitopes. Um, and one common feature which uh, occurs a lot is whether or not the residues have surface accessibility. And that makes a lot of sense because you cannot have an antibody interacting with residues which are not on the surface. But if you also just look at which amino acids are um, more prevalent, there are some patterns there as well. So tryptophan and tyrosine are more prevalent, and amino acids which are more commonly found in the core of a protein, they are more uncommon. Yeah. So this can sort of be your poor man's uh, method for predicting epitopes. But these tools that we're presenting, they use machine learning algorithms based on data to predict epitopes. And we distinguish between two types, those which are sequence-based and those which are structure-based. And the sequence-based just means that the machine learning models are trained on features which can be derived from a sequence. So they only require a sequence. And the structure-based ones are trained on features which can be derived from structure as well. Um, so that is, of course, a requirement. You need structure to use these sort of tools, but then these predictions are more precise. And one of the things that goes into the sequence-based prediction tools on IEDB are these amino acid scales. And you can think of these as features which describe different things for the amino acids of the antigen sequences. So hydrophilicity and also, of course, surface accessibility. And this antigenic propensity is just a measure of how often various amino acids occur as epitopes. And one or more of these amino acid scales goes in as an input to machine learning algorithms. Um, And the one which is our recommended one on IDB is a random forest algorithm, which is Bjoern. And the way this tool was trained, it uses these sort of features and then is trained on antigens where epitopes have been mapped from crystal structures. And then it's trained to predict these epitopes from non-epitope regions. So let's have a look at what the user interface looks like. Um, If you click here, it will lead you to the user interface where you need to provide a sequence. And if you have a Swiss product ID, you can provide that, or you can also just paste in the sequence. And then you can choose a method. And as I mentioned, the default method and recommended method is Bjoern. Um, and if you need more information, this is a hyperlink, and this will lead you to a condensed overview of how the tool works. But if you need additional information, you can go to the reference tab, and there will be links to the actual paper. So let's, if we input a sequence, this is a receptor binding domain from SARS-CoV-2, um, which we submit, and this is what the output page looks like. So on the x-axis, we have the amino acid positions, and on the y-axis, we have the Bjoern scores. And one thing we have to set is a threshold. So at what threshold do we want to predict amino acids to be part of an epitope? And by default, it's set at 0.5. And what this means that all these yellow areas essentially uh, will be predicted as epitopes. But this is a threshold you can adjust. If you want to be more sensitive or if you want to be more specific, you can set the threshold lower and higher. But at 7.5, it's these yellow regions. And at this cutoff, you can get the following predicted peptides as possible linear epitopes. And they're essentially just sequence segments along this amino acid sequence, which are above the threshold. Um, You can download the result set at that specific threshold as a CSV file, and this will also give you the raw scores, as well as a letter indicating which amino acids were predicted at as an epitope at that threshold. And we also recommend, so I mentioned that there were other methods available for predicting epitopes. And we also recommend that you check those out because if you can find sequence segments which score highly across all the different methods and there's sort of this consensus, this is more reliable than just relying on a single method. All right, so that's the sequence-based prediction tools. Let's move on to the structure-based tools. And the ones we're talking about are Discotope and Elipro. And as the name suggests, they require a structure, both of these tools. And the special thing about Discotope is that when it computes a score for a certain residue, it also considers the neighborhood of residues. So if you have a residue which is in a neighborhood of a lot of high scoring residues, all of these high scores will contribute to each other. And LA-Pro is an orthogonal method where it uses geometrical properties of structure. So it draws an ellipsoid onto the structure and then computes how various regions protrude the structure. And then the idea is if a region is protruding a lot, then that is a likely epitope. So the way Discotope was trained was it's trained on 75 X-ray crystallography structures of antibody-antigen complexes. And in all of these complexes, we look at how we compute like a log odds ratio of amino acids being epitopes and being non-epitopes. And this is our initial scoring, and then there's this neighborhood scoring on top of it. And then they also take surface visibility into account um, to get a final score. Um, yeah, so it's a structure-based tool, so you need to provide a structure. And if you know that your structure is something that is on the PDB, then you can provide the four-letter code. And if you just have a PDB file, you can provide that. Um, One caveat though is that you need to specify which chain you want to do your prediction on. So if you have a case where it's a multimer, our recommended solution right now is to just go inside the PDB and change all the chain IDs for each residue to be the same. Um, but to get a structure, you can go to the protein data bank, and here you see that there will be both experimentally determined structures around 200,000, and then there'll be 1 million computer structures, and these computer structures were computed with AlphaFold. Um, yeah, so to get a structure, you can use this sequence similarity search, where you paste in your sequence, and then you can toggle on whether or not you want to include computer structures in your search. So if you have an experimentally solved structure, it will look like the top search result. And if you only have the computer structures, it will look like the bottom one. Or maybe you have a combination of both. Let's first take a look at the experimental solved structures. For this, it's quite straightforward to get a PDB file. You can just click the download files tab and then click the PDB format option. For the if you only have sorry, then you also have to identify the chain, of course. And to do this, you can either just look directly in the PDB file, or you can just scroll down on the output page on PDB, and it will also indicate the chains which are in the file. For computer structures, it's a bit more complicated because um, very rarely can you download a PDB file, and uh, mostly you can only download a zip file, and sometimes I've seen you can only download the fastest sequence for some reason. But not to worry, there's always this link to the AlphaFold database, which you can follow, and that will lead you here, and here you can download a PDB file. Um, yeah, so the IEDB only right now only accepts PDB files, so this is why we need that. Um, yeah, so either way we can get a structure. And before we go to the output page, similar to Epitope, Discotope, um, for Discotope you need to actually set a threshold for epitope prediction. And under the Help tab, you can uh, find some guidance on how to set that threshold. By default, for Discotope 2, it's set at -3.7. But if you want higher specificity, um, you can set that at a lower threshold. So if we submit that, we should get an output that looks sort of like this. And yeah, so here the threshold is -3.7, which is the default. And that means that all the green areas will be predicted as epitopes. Um, but this can be adjusted. And you can save your predictions as you adjust your threshold as a CSV. And you can also just have a look at the data or the predictions in the web page by clicking the table view. And this can give you an overview of the scores and also other useful information, which is this contact number. And contact number is an indication of how buried various residues are. And this green colored rows indicate which residues were predicted at epitopes at your threshold. We also have this 3D viewer, which allows you to visualize the epitope that you have predicted. So moving on to ELIPRO. So ELIPRO, as I mentioned, uses geometrical properties of the protein structure by approximating a ellipsoid onto it and computing how various regions protrude in this ellipsoid. And it's interesting and cool because it also gives you both linear and discontinuous epitopes on the output page. Um, but otherwise, it has a similar input page as Discotope. You also need a structure to do predictions, and then it has this minimum score, and this is similar to the threshold that we had earlier. So this essentially means that at what minimum score should we predict something as an epitope. And so this is something you can adjust uh, to adjust sensitivity um, or specificity. Um, there's another interesting option, which is this maximum distance angstrom, and that's because ELIPRO gives you discontinuous epitopes, so full sets of residues which constitute an epitope. And this is a restraint on how far away those residues are allowed to be in that set of residues. So, oh yeah, and we also have an issue here for the multimeric complexes. So, the way that the ELIPRO works, it will consider if you give it a multimer, it will make predictions per chain. And so, if you want the model or the ELIPRO to consider the entire multimer at the same time, you need to go into the PDB file and change um, all the chains to have the same ID. Yeah. Yeah, and when you run the prediction, you can also, if there are multiple chains, you can select which ones you want to make predictions on. So this is the output page, and as I mentioned, you have both the predicted linear epitopes and the discontinuous epitopes. So the linear epitopes, these are all uh, amino acids which are next to each other in the sequence. And then the discontinuous ones, they're not next to each other, but they're within this 6 angstrom distance that we set. And we also have a 3D viewer for ELIPRO, allowing you to visualize your predicted epitopes. And there's a graphical output page as well, um, which shows the predictions at your set threshold. Um, yep. So that concludes the tools and examples. Now we'll talk about some up and coming tools. Um, and since these ELIPRO and Discotope and Pepper were released, these versions here, a lot has happened in the field. And one of the things is something that I've worked on at DTU. So this is Pepper 3, and this is currently only available on DTU, but we're working on creating a nice user interface for this on IEDB and incorporating that there as well. So Pepper 3 is similar to Pepper 2 in that it's a sequence-based tool, but here we incorporate evolutionary features as part of the model. So um, evolution of proteins can encode a lot of useful information. So if we see that compare two proteins which we know are related evolutionary, and we see that some amino acid pairs mutate at the same time, it's an indication that they are in contact in the protein structure. Um, and also you find amino acids which are conserved, so never change across all of your evolved proteins. And it's something like a lysine, which is a special amino acid which allows for a lot of backbone flexibility. It's an indication, or it could be an indication of an active site. So that sort of information is something that we incorporate in Pepper-free. And the traditional way to get this sort of information, um, and still is a very good way to do it, is to create multiple sequence alignments and sort of create a lineage of all your protein sequences. And but a couple of years back, these large language models were showing that if you trained um, on these millions of sequences, protein sequences that we have, you can obtain a lot of the same information that you'd normally need to make a multiple sequence alignment for. And we use one such pre-trained language model called ESM2 to get a large boost in performance. Um, and then there is another tool. This is essentially the next version of Discotope, which is a structure-based tool similar to its predecessor. And this work was done by a former colleague of mine, Magnus Høy. Um, and he used a deep learning approach called inverse folding. And inverse folding is the opposite of what AlphaFold does, where AlphaFold tries to predict um, the atomic coordinates of a sequence, the structure. Inverse folding does the opposite. Here you have the structure, then you try to predict which amino acids would fit into this structure. And inverse folding, or sorry, ESMIF1 is this pre-trained inverse folding, which is trained to um, predict these amino acids which fit into structures for like a lot of predicted alpha structures and solve structures. Um, and right now, these user interfaces are under development, and this is something that hopefully will be released in the first quarter of 2026. So to conclude, I hope that you can understand that there are a couple of B-cell epitope prediction tools on IEDB, and I hope that you understand how they were built and how you can use them. Um, Otherwise, I'm happy to take any questions that you have. Thank you.
Awesome. Thank you so much, Joachim, for that wonderful presentation. As we have a few more questions, I'll give everyone an opportunity to submit a few more questions. I'd just like to reiterate that obviously much of this presentation, in contrast to the first half of the session, where we were focusing on next gen, as you can see, a lot of the B cell tools are still on the classic site, but as you saw in those last few slides, they are actively under development to be implemented on the next generation platform. So it's always cool to be able to show our users what's coming and what's behind the scenes. So hopefully by the start of next year, we will have transitioned the majority of our the big classes of class one, class two, and B cell epitope prediction to next generation, which will be really nice for everyone. So now we will move into the section 3 Q&A, and I'm inviting Bjorn back to help with the questions for Joachim. So
Okay.
Bjorn, did you want to take a first question?
No, that would be
I'm sorry.
unfair.
I can't. Where the I can't see. I don't can't see the
If you
questions for some
if you
reason.
click on the Q&A,
Yeah, it just says.
there
Wait, are there
like
maybe I can only see two questions right now.
they're
Oh,
popping in right now.
okay, okay, okay.
Um, Actually, I mean, okay, I'll just take the first one then. The first question is from Jacqueline, and the question is there a way to include post-translational modifications? And Joachim should correct me, but as of now, there isn't. I think the
Yes.
same as for T cell epitopes, and actually even more so for antibody um, epitope recognition. Very clearly, there's a major role of post-translational modifications. I mean, the role of glycans for HIV protein recognition or so has been very well documented. Um, so at the same time, it is extremely hard because the data sets of these it adds just like a dimension of diversity and complexity that we don't have represented in our training data enough to really make good new predictions that take them into account. So we are not
Yeah,
saying that they're irrelevant, but they are currently not dealt with by the algorithms. Sorry,
yeah.
and yeah, this okay, if that was fair. You want to take the next one?
No, yeah, sure. I guess I'll just take the next one, which is from Mauricio Calvo Calle, and it's how good are predictions for B-cell epitopes? And I would say it's quite a bit worse than it is for MHC peptide predictions. Um, and it has one thing is that we don't have as much data, of course. A lot of what prediction, like all these tools, like WebPred, they're trained on all of the solved structures that we have, which is sort of the gold standard for a B-cell epitope. And there's just not a lot of it. And then there's other issues of when we're training these models, a lot of these antigens that we have data for are understudied. So um, I may have an antigen and then I only have a single known antibody interacting with it. And then uh, I'm training the model like for the remainder of the surface of the antigen. There may actually be an antibody that interacts with it, but I just don't know. So when I'm training the model, I'm training it to predict negative on this entire area, but in reality, it's sort of unknown, right? Um, so it's both hard to evaluate these tools and yeah, and hard to say if they're good. I think that the best way to evaluate them is like look at like really prototypical antibody targets like lysozymes where we have almost the entire surface covered, and then and then see how well it is able to predict that. Yeah. Anna, do you have anything to add, Bjorn?
No, I think that was
All right. Yeah, I think
uh,
if we had to give a number, I think the AUC is like so for before Pepper 3 it's around 0.6 or something, and then now the AUC is 0.7. and it's been that way for a while. And I think it's hard to move the AUC higher because of this problem with understudied antigens, I think, right? Um, yeah,
Thanks.
yeah,
I will take the well this oh I shouldn't do this. Okay, this was it says very much for you. Um okay, actually there's sorry. Joachim, these are all very much in your ball house. Can you
yeah.
maybe take the next
I
one?
see that. I see that. I should have given you the one I just answered. That one was pretty good. Uh, right. So no, 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 no. Yeah. So there's one here called how are the new tools better than just running AlphaFold? Um, so I think that what you mean here is that if you have both the antibody and antigen, so then you would want to predict that specific interaction, right? 'Cause it's not just running. So um, I think that is a more interesting task, and that's actually what I've been using most of my PhD on doing is trying to map the specific interaction between an antibody and antigen. And so, but it's sort of an entirely different problem. What we're doing here with these tools is trying to predict all likely B-cell epitopes given an antigen, um, not for one specific antibody. So these sort of tools that we have here, they could be important if you want to assess, um, I don't know, the risk of a molecule, like if it could elicit an immune response from the B cells. Yeah, so it's sort of uh, a different thing than just running AlphaFold, these tools. Do you have anything to add to that, Björn?
No.
All right. Um, then there's one about this is from an anonymous attendee. I was wondering for ESM, which parameters did you use? Um, so that's for Web free. God damn. And they have a number of different. I think I used the one that has 650 million parameters. Because they have a number of different sizes of ESM, um, which are faster. And then the smaller, as the smaller they are, the faster they are, and then you lose a bit of information in the embeddings. Um, and then I train a neural network on top of these embeddings. And the neural network we found to work the best was just a feed-forward neural network. We also tried incorporating or using convolutional neural networks and long short-term memory cells. Uh, but I think the embedding itself is quite informative already, so you don't really have to um, put on a very complex neural network to make or to get a good performance. Um, yeah, I hope that helped. Um, is there any question you would like to take, Björn? Or should I just
This is, I mean, some of these are redundant with what you already just answered.
okay,
Uh, at the bottom, there's a question. Maybe I can try to take that and you can weigh in. Like, um,
sure.
what is the computational cost of the new models?
Uh,
So, I mean, there's like two different parts to the cost. One is anything that is needed for the method development, where we have to create a lot of models and uh, optimize parameters and trying to find like optimal parameters across like a large possible space that is extremely computationally expensive.
mm,
Then as things are being run, the computational costs obviously mean on purpose we are trying to not implement things that we can't execute. So there's always an attempt to keep things
mm,
doable. So, I
Mm.
mean, I don't know, Joachim, you want to answer that better?
Um, uh, yeah, this is like the Prepper 3 is quite a deal slower, or is quite a bit slower than Prepper 2 because here we're relying on a language model. Or Prepper 2 was these sequence windows of amino acids, like one-hot encodings and some other features that we put in, like server accessibility, and that's a lot quicker to compute than make language model embeddings. But we can run these language, we can create them on when you run it on GPU in batches. It's not too bad. Um, I would say we're running it on the health tech server on DTU, so you can test it there and you can try for yourself um, how slow or fast if you feel it is. Yes. Um, uh,
Okay, I think, I mean, what I'm seeing is the same questions as we've had before, which clearly means B-cell epitopes are so easy to predict that um, and you, yeah. Nina, if you want to come back on, um,
I
I
think
think,
maybe
or,
I'm not sure if
or
that.
did you want to answer another question?
Nah. I just wanted like this one from anonymous entity. With AlphaFold, it becomes easier to use structure-based methods, but is AlphaFold precise enough to define epitopes with high confidence? Um, yeah, so I mean, I guess like AlphaFold. Wait, no, I think we
I thought you answered that before, right?
yeah,
So
yeah. I think I sort of answered that. Yeah,
yeah. So the
yeah,
best part about things like AlphaFold is let me try to rephrase that also all
yeah,
these advancements are extremely useful to be able to use existing methods that require a structure of the antigen, where
yeah,
in the past there was a major limitations and we didn't have good ones. And
yeah,
we had like long guidelines on how to create your own homology model of your own antigen. Now I would say definitely screw that. Use AlphaFold and all the likes to get a good model of your antigen. But then using and then there are attempts of doing whatever, adding two proteins and giving the docked version out with antibodies. They just don't work well. If I mean that's essentially what Joachim has been working on to show that a naive use of something like AlphaFold to create like a docked structure of an antibody with an antigen is not a reliable thing to define like high precision epitopes.
uh,
Is that fair?
I mean, it's I mean it's not I mean it's we sort of like in my paperwork work we did I mean we use AlphaFold sort of the tool to do the mapping between antibody and antigen, and then we show that, and that AlphaFold has its own confidence score, and that can sort of get you quite far, I would say. I mean, compared to earlier docking methods, but it's still very hard for sure, right? You're muted,
So
Bjorn.
sorry, maybe we should have gotten into those details yet, but like in your work, you were showing if you were just naively using it without any thoughts, you would
Yeah.
get like one model back. And so I mean, you did a lot of work to like force even sampling the space to get like a good repertoire of antibody antigen
Yes.
configurations,
Yes.
etc. All of this obviously is not implemented on the websites, as I guess we didn't talk
No.
about it
No.
here, but there's like a lot of work trying to get
Yes.
into it. If you naively just put the two things in and run like a standard off-the-shelf AlphaFold prediction, the likelihood that you're going to get a novel epitope correctly predicted back is practically zero.
Uh. Yeah. Yeah. Yeah.
Awesome. Thank you so much, Joachim. Thank you, Bjorn. I think we've answered every question available. If there's anything else, we can add a type response, but I think we covered everything thoroughly. So thank you both so much.